So this video follows on from the previous one, which covered autosomal recessive inheritance, and this will now cover autosomal dominant inheritance and also incomplete dominance inheritance from that Unit 2 section on reproductive biology, covering ante and postnatal screening. So this first example here I'm going to cover will be autosomal dominant inheritance, and it's very similar in fact to the recessive inheritance, except that a condition will be passed on the dominant allele, whereas previously we talked about it being passed on the recessive allele. So in this example then, if a person is affected, they could either be heterozygous, okay, and as an example then I'll put capital H, small h, or they'd be homozygous, dominant, and therefore have two capital alleles. So this time now, because that condition is passed in the dominant allele, if you are heterozygous or homozygous dominant, you would be affected. You would only be unaffected if you do not possess any of those dominant alleles. So therefore, if you're recessive, okay, if you have two recessive alleles. So an example of this disease, or a disease that would be carried by this type of inheritance or passed by this type, would be Huntington's. Full name would be Huntington's chorea. Now, a person that's affected must have had at least one affected parent, because if they didn't, therefore then the parents must have been recessive and they would never have been passed that gene. So a person affected must have one affected parent, and that's at least. It could be both, but we must have at least one. Now in terms of this this pedigree chart, same applies as to the one previously. So circles would be females, squares would be males, but in this point it doesn't really matter. And again, this would be your P generation, this would be your F1, and then this would be your F2. So in this cross I've given the letters that I'd want to use for a Huntington sufferer, and that'd be a capital H. And the unaffected people would be a lowercase h. So their allele would be a lowercase h. So for this case, rather than the recessive ones being affected, like in the previous example, all the people that are unaffected are recessive. So all the white boxes and white circles would be recessive and have homozygous recessive as their genotype. And every other single person on this who has Huntington's must have at least one capital H to have inherited that condition. So each of these people would have a dominant allele. And then in the same procedure as we did before, you would work your way back. So in this example, there are two recessive alleles here. This person is homozygous recessive, which means they must have got one recessive allele from one parent, so the other one must have come from the other parent. And that same logic is applied here. Now this child must have inherited that dominant allele from this parent here, and the other parent can only ever give a small h, so therefore would have inherited the small h, the recessive allele from that parent, and therefore would be heterozygous. So again, if we work our way back for this family, this person is homozygous recessive. They must have got one recessive allele from one parent, the other one must have come from the other parent, and therefore that parent is heterozygous. Same logic applies here. So you have one homozygous recessive child. Each small h must have come from each of the parents, so these parents would both be heterozygous. In terms of working out their children now, we don't know whether each parent gave a dominant allele and therefore would make these children homozygous dominant, or one parent gave a dominant and one parent gave a recessive, so they could be either homozygous dominant or heterozygous. Now again, a question may ask, if parent A and parent B decided to have another child what would be the chances of that child having Huntington's? So again, you would do the Punnett square just like in National 5. Always make sure you do this because it's easy to make mistakes. So parent A, I'm just going to pop at the top. Parent B, I'm going to put at the side. If you do the Punnett square like normal, this person would be heterozygous. This would be homozygous recessive. This one heterozygous. And this one homozygous recessive. So if you look at the 4 potential genotypes, there's really only 2 because they're both, each of them the same. This person and this person, or this potential genotype and this one, would be Huntington's. And these two would be homozygous recessive and therefore unaffected. So this person and this person, potential genotypes here, are 50% of the whole lot. So 1 out of 2, or 50% chance of inheriting that. Now the next example is incomplete dominance, which kind of goes against what we've been talking about all the way through National 5 and also into the beginning of higher, as it works slightly differently. So incomplete dominance is actually sometimes easier than the other crosses because they have to give you so much information to be able to work it out. So an example of incomplete dominance would be sickle cell anemia, which is what is here. And I honestly haven't seen another example in exam questions, so it's typically that one that would be shown. There are 3 different phenotypes, potentially. Now normally we would have 2 different phenotypes, so black hair or red hair in a cross. And therefore, because there's 3 different phenotypes, there's also 3 different genotypes. Now in this, normally we talk about dominant being a capital and recessive being a small, but in this example there are no recessive. There's just a mixture of 2 different dominant ones. So in this, if you have normal hemoglobin in your red blood cells, you would have 2 capital H's. If you have sickle cell trait, you would have 2 capital S's. But if you have what is essentially a mixture of them both, you would have a capital H and a capital S. Any examples they would ever give you, they would have to give you the genotypes, and therefore you would always use this. So in these examples, it is really straightforward to carry out a cross, or to complete a pedigree chart on their genotypes. So again, P generation F1 and F2, and you just look at the key to determine who has got what in terms of the genotypes. So every white circular square would be capital H, so homozygous dominant. So all these white circles or squares would be homozygous dominant for each. All the ones that are coloured in black, circular square, would be 2 big S's. And then the ones who are a mix of the sickle cell trait that are cross-hatched would be then HS. So because this is so straightforward, normally the questions would revolve around asking what would happen if parents had another child, much like in the previous example. So if I say that parent A and parent B wish to have another child, the question might ask, what is the chances that their child would then have sickle cell disease? So again, you would just have to do the Punnett square. So parent A I'm going to put on top and parent B on the side. And you do the cross. I know this looks slightly different because the letters are different, and it helps if I do it correctly. But it still works the same way in terms of pulling the letters down. So I was doing it right, never mind. So I'll fix that again. So 2 capital H's here, heterozygous here, heterozygous here. Okay, we're pulling the H down and the S across, and then this SS here. So the question asked, what is the chances of their child having sickle cell disease? Sickle cell disease has 2 S's. So right here, out of the 4 potential genotypes, or 4 combinations, there's a 25% chance of them having sickle cell disease. There's a 50% chance of them having the trait, and a 25% chance of them having normal hemoglobin. Okay, again, same rule applies, so you could do the same with this parent and this parent, just to make sure that I show you a correct Punnett square, where I don't mess up. So this parent at the top, HS, and this parent on the side, SS. So you would do your cross as normal. And again, they could ask, what's the chances of any other child they have having sickle cell disease? Out of the 4 potential genotypes, there are 2 different ones. So 50% of those would have that sickle cell disease potentially. Okay, so I'm going to stop there and I'll move on to the sex-linked inheritance conditions, which are the last ones.